يعطيكم العافية يا شباب وصبايا. أول شيء بدي أتشكر هيدي السجشن كرمال هيدا الفيديو and let's get started. بهيدي الليكتشر بدنا نكون عم نحكي عن الفولدing of proteins. We're going to be dividing this video into two main parts, folding of small proteins and the folding of large proteins. نبلش بالfolding of small proteins and it's quite easy. نحن عندنا two main steps. أول step هي formation of the secondary structure and this is very fast, and the second step is the formation of the tertiary structures, which is the folding of the secondary structures, and this is going to be taking a little bit more time. As this process is happening, هيدي الfolding may sometimes be inaccurate, and just هيك as a side note, يعني نحن ممكن أي بروتين بدنا نعملها folding بكذا طريقة، بس only one type of fold is going to be giving us the operational protein. So the folding of proteins is extremely important to make sure the protein is operational. نكفي نحن هلا على what could happen يعني إذا نحن كان عندنا incorrect folding of these proteins. أول شغلة نحن عندنا two states. أول state اسمها molten globule state. هيدا molten globule state simply it is a protein that has been folded but it is unstable and it is unoperational. So هيدا ما كتير بتعمل stress للسل عشان it is immediately going to be breaking down and the protein is going to be folded again. So ما في أي مشكلة. Now the second part is called the discrete folding intermediates. هيدول discrete folding intermediates مثل ما نحن كنا عم نحكي بالكيس اللي قبلها, the only difference between this and the one before is that this is stable. So هيدا protein is not going to be disintegrating, it is not going to be breaking down easily. Therefore نحن صار عندنا هيدا protein that is non-functional and also it is not breaking down. So an accumulation of these proteins رح يكون عم يعمل stress كتير على هيدي السل. نفوت نحن هلا على folding of the large proteins. It is quite different. هون نحن by the way عم نحكي specifically in bacteria. So عندنا نحن two main proteins or two main chaperones that are going to be helping us. هيدول by the way chaperones equals helpers. These are going to be helping us folding these large proteins. هلا الاثنين في وحده اسمها HSP60 and the second is called the HSP70. هلا just follow up on the HSP70. It is going to be binding to the protein that is going to be folded and it is going to maintain its linear structure. هيدي الشغله كثير مهمه عشان او بالاحرى خلينا let's picture a scenario. انا عندي هيدي البروتين it started folding up on top of each other ونحن بدنا نرجع نعمل لها folding مره ثانيه بال HSP60. It is not very effective, it's not very efficient. So in order to maintain, in order to get the highest efficiency rate, نحن عندنا هيدي ال HSP70 protein that is going to be binding على البروتين itself and maintain its linear structure. And also هيدي ال HSP70 عندها another function which is guide this protein to the HSP60. هلا ال HSP60 لحط هلا صوره هون. في نحن عندنا the HSP60 as the structure تبعتها is quite easy. It's like two donuts stacked on top of each other. فكرتها انه هيدي البروتين اللي هلا by the way هي linear رح تنزل جوات بين هيدول الدونتس and within this HSP60 chaperone it is going to be, the protein is going to be holding onto the walls of this of protein and then disintegrating and then detaching. So attaching and detaching continuously until it is folded correctly وبعدين رح تطلع من ال HSP60 completely folded. هلا بس في شغله صغيره انه within the HSP60 في نحن عندنا a structure the cap that is called the GRO-ES. هيدي ال GRO-ES عندها two main functions. اول شيء maintain the hydrophobic environment that is present within the HSP60 and also it is going to be slightly enlarging the HSP60 protein in order to make sure the protein is getting folded correctly.